Hello everyone, and welcome to today's webinar, which is entitled Challenges and Considerations for mRNA Therapeutic Development. Now, this is the second episode in our series of 4 webinars, being brought to you by Jen and the CRISPR Journal, that will cover some of the latest advances in RNA research and technology. This webinar series is being made possible through proud sponsorship from TriLink Biotechnologies. TriLink Biotechnologies, part of Miracco Life Sciences, is a CDMO for overcoming challenges in the synthesis and scale-up of nucleic acids, NTPs, and mRNA, including its proprietary CleanCap mRNA capping technology. TriLink offers cGMP and general mRNA, oligonucleotide, and plasmid manufacturing to support therapeutic, vaccine, and diagnostic customers. I'll be your host for today's event. I'm Jeff Bogaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which has been at the vanguard of the life sciences industry for more than 40 years, bringing you the latest information on the newest tools and technologies that can aid your research endeavors. Now, using RNA as a therapeutic agent is not a new idea that sprang up just recently in order to fight SARS-CoV-2. Investigators have been postulating and conducting research to use RNA as medicine for more than 30 years. The COVID-19 pandemic provided an opportunity for RNA-based therapeutics to finally break through to the clinic. In our first webinar of this series, we heard how RNA can be used to treat other infectious diseases like influenza. Now, if you haven't seen that webinar, make sure you check it out. The link's in our resources section. In today's event, our distinguished guest speaker will take us through his extensive experience with mRNA chemistry, sequence design, and commercial scale production, as well as the challenges he faced along the way. But before we begin, let's first get to know a little bit more about our speaker for today's webinar. I'm delighted to welcome Dr. Edward Miracco, who is the Director for RNA Chemical Biology and External Technology Investments at Moderna. Ed has been interested in RNA, its chemistry, and its function for more than 20 years, and it was that curiosity that led him to pursue his graduate degree that focused on investigating the mechanistic enzymology of the pseudouridine synthases. And as a postdoc, Ed continued with his affinity for pseudouridine and was able to solve the first complete structure of a telomerase holoenzyme by cryo-EM, all the while simultaneously keeping abreast of the seminal work from Katie Miracco and Drew Weissman, which showed that pseudouridine containing mRNA dramatically increased total protein produced while decreasing the reactogenicity from exogenously delivered mRNA. Now, it was the culmination of all this work that convinced Ed that mRNA therapeutics were poised to unlock a new frontier in medicine. Ed jumped at an opportunity to work at Moderna in the mRNA process development group and has been forging new ground ever since he started there almost 9 years ago. Now, today, Ed's going to take us on an exciting trip through some advanced mRNA biology and chemistry, as well as give us some insights into how mRNA is being used therapeutically and novel ways it could be utilized in the future. But before we get to Ed's talk, first, I want to remind the audience that we'll have a Q&A session right after the presentation ends. Ed's going to be on hand to try to tackle as many of your questions as he can. So if you have a question for Ed or about his presentation, simply type your question into the Q&A box and hit submit. You can do this at any time during the presentation. You don't have to wait until the end. If you have a question and it pops in your head, just type it in, send it on in. Okay, now that we have that all out of the way, I'm going to hand things over to Ed.
Hi, my name is Ed Miracco, and I'm the Director of RNA Chemical Biology and External Technology Investments at Moderna. Today, I'm going to talk to you about an introduction to mRNA as a medicine, specifically from biochemistry to pharmaceutical science. As Moderna is a publicly traded company, I have to put a forward-looking statement and disclosure slide. This basically says that we are going to talk about science, and sometimes things don't work out as expected. So we'll jump right into it. The information flow in a cell typically goes from DNA to mRNA to protein. And so, the way it's drawn out here is linear, but realistically, there's lots of feedback loops. But for the sake of discussion here, we'll assume it goes in one direction. So you typically start with DNA, where the information is stored kind of as a cold storage process, right? You want it to stay very safe. And through a process called transcription, that DNA is read and turned into mRNA, and then that mRNA is then read and turned into protein. And so to kind of think about this, I like to talk about analogies, and I like to think about Star Wars a lot. And so if you are familiar with Star Wars, this is a Star Destroyer. And one of the interesting things about Legos is that you can typically remake them after you've built them the first time, as long as you don't have kids destroying the instruction manuals or dogs. And so basically what I have here is an example of the instruction manual that's in, perfect, pristine copy. And this is what we'll like to describe the DNA as. But because you want to actually make this over and over again, you make a photocopy of it. And that's a lot like the mRNA, right? You don't care if it gets destroyed. You could mark it up, you could rip it, you could throw it away. Basically, the idea here is that it's disposable. So you've got your DNA copy. This is like the cold storage. You want it to stay pristine. You want it to not lose any information. You photocopy it. You have a disposable copy. You don't really care so much. And then that basically gets turned into the Lego Destroyer by you, right? And so in all of these steps, you have different parts of the cell that actually do the different steps of this information transfer. And so in the biotech field, there have been many companies since the discovery of DNA that have basically leveraged the power of proteins to make medicines, and also basically managing DNA to use for different uses, from everything from crops and food production to science in molecular biology. And so the use of DNA, and then of course the use of protein to make medicine, conspicuously skips past the mRNA. And so, I like to put down crickets here. And as I've been in the field now for a few years, it's been interesting following kind of all of now the companies that have popped up, right, to leverage mRNA as an actual modality for a medicine. And so this has been really fascinating to watch, especially with the COVID vaccines coming out, primarily being mRNA-based. The power and the ability to use mRNA as a medicine really has been suddenly realized. So let's talk about DNA. So DNA is a magical compound. Really, in all of our cells, you have two kind of strands that come together. And these two strands meet in the middle, and these parts that meet in the middle, the bases, pair with each other in very defined rules. And so this base pairing process that DNA uses allows you to actually separate the two DNA strands to replicate them perfectly. And so this is how cells divide, right? So you've got your perfect double-stranded DNA, you separate the two strands, you fill in the opposing or the missing strand, because based on the rules, A base pairs with T and G base pairs with C, what's missing when you separate the two strands. And so this has also the added benefit of putting the information part of the molecule kind of tucked away inside this double strand. And so it's very important for fidelity, for keeping the DNA, in one piece, to actually dehydrate it. And so it's really important that the double-stranded DNA actually is able to exclude water from the inside portion of it. And so how do you actually then make the mRNA from the DNA? Well, here's a little video to show. So you basically have an RNA polymerase. In this case, it's in purple. It's this purple blob here. And you have incoming ribonucleotides, or NTPs, that base pair through those rules, right? A to T, C to G, but in RNA it's U. But basically these base pairing rules come in and allow you to actually make an mRNA using the DNA strand as a template. And so you can see here, the DNA is barely opened up a little bit, right? Because you want to protect it inside the cell. So it's barely opened up a little bit, and the mRNA strand is coming off of that DNA strand, using it as a template. This is the photocopy step, if you kind of think back to our first slide. And so here's a 10,000-foot view of the same reaction. Basically, you've got your RNA polymerase in blue or cyan here, racing down this double-stranded DNA. And this is a beautiful video. It shows kind of the crystalline nature of DNA, right? It's this nice repeating structure. And then out comes the unruly cousin of DNA, RNA, that's just this tangled mess. And I like to think about it like headphone wires in a pocket, right? It gets tangled up into knots. And so here are the incoming nucleotide triphosphates getting stitched together by the RNA polymerase when they properly base pair with the DNA template, and off goes the full-length mRNA. So how does the mRNA then get used by the cell? Well, through a process called translation, a machine in the cell called the ribosome actually reads the sequence in the mRNA to turn it into protein. And so the mRNA has a few components that I'm going to talk about now, and then I'll go into more detail later. So on the left-hand side of the mRNA, which was typically drawn as the 5' end, and that has to do with how the atoms are lined up, but basically the left side is the 5' end. The ribosome and its initiation complex components bind this cap area called a 5' cap, which we'll talk more detail later, and they recognize that, and they utilize this 5' untranslated region, or 5' UTR, as a landing pad, because the ribosome is a big machine. It's about 40 nucleotides in width total. And so if you don't have some buffer region here that prevents the ribosome from missing out on some coding sequences, you run into problems. So the ribosome comes here, finds the cap, sits down, it has this buffer of the 5' UTR, and then it starts scanning along for AUG, the triplet codon, those 3 nucleotides. And this is the start codon. And so these are far enough away from the cap so that you don't run into jumping ahead and having truncated proteins produced. And so you find this AUG, and the ribosome says, okay, good, this is where we start reading this coding sequence here. And it turns this sequence into protein. And so through this process called translation, you elongate the protein, and it then runs into a stop codon, which is at the end of the coding sequence, which is also followed by a 3' UTR and a poly A tail. We'll talk more details about the 3' UTR and poly A tail later, but basically the 3' UTR is a regulatory element, and the poly A tail is also part of this whole complex. So at the end, you terminate, you release the functional protein. Okay, so let's dive into the ribosome a little bit. So translation is the process by which an mRNA is translated from the language of nucleic acids to the language of proteins. And so the way that happens uses adapter molecules called tRNAs, or transfer RNAs. And these are like decoder rings. And it's like a Rosetta Stone almost, where you basically have these nucleotide triplets in the mRNA. Through base pairing rules like we talked about earlier, like A to U, C to G, through those base pairing rules, these tRNAs recognize these triplet codons, and on the end of the tRNA is a single amino acid. So like an area code refers to a area in a country, right, for phone calls, which is so different nowadays with cell phones. But back in the day, long distance phone calls were very important to know the area code from where you were calling. This amino acid is associated with these 3 nucleotides. And so this tRNA binds the mRNA through base pairing rules, and if it's a good fit, it moves over to the next site and hands off the amino acid to the growing protein chain, and then eventually the tRNA is released to be recharged by the cell and reused, and having the same amino acid added back to the end. So here's a quick video of translation to show what it looks like. And so basically you've got your 2 subunits coming together of the ribosome. They read the mRNA in these triplet codons. They ratchet through 3 at a time. And these little green L's floating around have little red balls on the end representing amino acids. They come in, they base pair to the mRNA. They donate their amino acid to the growing protein chain, and they then get released into solution to be recharged. Now, here's a great video example of a protein folding while it's being made. And this is really important. When a protein folds while it's being made, it folds into a structure that's unique for what the context of the amino acids are at that moment. And so if you were to say, unfold this protein after it was made, it will not necessarily refold into the same structure, because the context of every amino acid with the surrounding amino acids actually will prevent it from forming the same structure. So imagine you have a bunch of positive charges on the end of the protein and a bunch of negative charges on the beginning of the protein. Well, when you're making those negative charge portion of the protein in the beginning, they're going to fold up in a way that, maybe isn't ideal energetically, but that's the best that they can do. If you then unfold that protein after it's made, those positive charges are going to really want to interact with those negative charges, and you're going to misfold. You're going to unwind the protein and let it refold in a way that is not functional. It's a denaturing event, and it forms aggregates typically. So anyone who has ever cooked eggs knows that when you have the egg first cracked, it's liquidy, it's clear. The egg white is actually liquid, it's clear. And so if you then heat it up, you unfold those proteins, they actually aggregate into egg white and become semi-solid, right? And so that's the concept here of proteins folding while they're being made form a specific structure. That is completely different from an mRNA. And so let me talk about those details here. So we've got our mRNA structure, or primary sequence here, our schematic. We talked a little bit about the 5' cap and the 5' UTR, and then the coding sequence, right? This green sliver here is the start codon, the red sliver is the stop codon. After the stop codon is the 3' UTR. The 3' UTR in cells is typically used for modulating mRNA stability and protein expression. You've got a lot of microRNA binding sites in the 3' UTR that change how long an mRNA survives in a cell. It also changes different cell type expression profiles. So it's basically regulatory region, regulatory elements. After the 3' UTR is the poly A tail. Poly A tails are typically a string of 100 A's to 150 A's. And they are very important for stability, and then to also form a circular mRNA structure with the initiation complexes. And so the most important point here to mention is that if you break an mRNA, if you fragment it in the middle, you actually wind up with a piece that has a cap and no tail, and a piece that has a tail and no cap. Well, neither of those pieces will actually make protein. So by definition, you need a cap and a tail in the same mRNA in order to actually successfully make proteins. So this is a nice built-in kind of safety feature of using an mRNA as a medicine, because it means that if you have a degradation event, you're not making truncated protein. And that's really important, because truncated protein can actually lead to bad problems in safety and lack of efficacy. And so that poly A tail at the end, as it turns the mRNA more into a circle, allows for translation to occur effectively, and you need these signals. And so the point I was mentioning before about protein structure forming while it's being made, the really cool thing about an mRNA is that its function is actually derived from its sequence and not from its structure. And this is really important for mRNA manufacturing, because one of the ways that you can actually clean up an mRNA is by unfolding it and allowing it to refold after purifying it. So as long as you don't result in fragmentation or bond breakage in the mRNA, you can unfold and refold it as many times as you want. And this allows for really stringent purification methods, which is actually very important, especially in the early days of the field, of making a safer product, because there are impurities that really bind tightly to the mRNA. And the only way to remove them is actually have high temperature and solvent-based methods for purifying it. So let's talk more about how an mRNA is actually used in the cell and how it's actually can be used as a medicine. So what we have here are two different cap structures. We have cap 0 and cap 1. And so cap 0 has a hydroxy group at this N+1 nucleotide position. And what you'll notice here in the cap structures, you have a 5' triphosphate and then an inverted 7-methyl G. So we'll talk more about the cap structure later, but basically the point of this slide is to show the cap 0 versus cap 1 difference, whereas in cap 1 you have a methyl group. And so I put a couple references over here to talk about how cap 0 is actually a handle for viral infections to be recognized by the cell. So most viruses can't put a methyl group here very easily. They're able to actually put caps due to encoding their own capping enzymes, but this is a harder position to modify. And so most viruses don't have that. And so the ability to differentiate self from non-self using this cap 0 versus cap 1 difference is very important for your cell to avoid viral infection. And so there are a lot of early discussions in the field around, can you use cap 0 versus cap 1 for an mRNA medicine? And so in order to actually manufacture an mRNA, especially in the early days, you used an enzyme that actually adds the cap post-transcriptionally. And so in order to add the cap1 methylation, you have to have a whole nother enzyme. And so having all of these, different enzymes and different reactions in your mRNA capping reaction is actually very difficult to control, right? So if you have a lot of the inverted G added, but not a lot of the methyl added for cap1, that's not a very good product. And so it's important to keep in mind how hard it is to do different unit operations when making an mRNA medicine. And so, it turned out that when you wind up going into higher order organisms, specifically rodents and then into non-human primates and then into people, the cap1 methylation turns out to be very important for getting the most protein made per mRNA delivered. And so this kind of total protein produced per dose is a very important attribute for making an efficacious mRNA medicine. So how does the cap actually work? How does it initiate translation? So as a structural biochemist by training, I tend to think a lot in structures. And so here's an example of the EI4E protein, which is part of the initiation complex. And what you can see here tucked in is a cap analog for the co-crystal that actually mimics the N7-methyl-G triphosphate or inverted 7-methyl-G triphosphate of the cap. And you can see how it kind of tucks in really well into this binding pocket. And what I'll show in a second, this planar amino acid here and this planar amino acid here are two tryptophans. And so, one of the really cool things about this binding is that it requires this positive charge to actually make a pi cation stack. So these two tryptophan residues actually stack with this positively charged N7-methylguanine. And this is how you actually bind effectively EI4E and allow the whole thing to kind of start translation. And so if you don't have this methyl group, you don't have that positive charge, you don't actually get high binding affinity. And there's been some experiments done where you actually deliver cap-G, so without that N7-methyl, and you get a little bit of translation. It actually turns out that you actually get some methylation in situ when you deliver just a cap-G mRNA. So it's a very fascinating story of how delivering an exogenous RNA actually leads to weird things in the cell, and different types of reactions that would have normally, say, happened to the nucleus tend to happen a little bit in the cytosol, and you wouldn't have detected that unless you had kind of delivered something abnormal. So the other thing that the 5' cap does is actually protect it from degradation. And so inside the cell, this is a really important part of the cellular biogenesis. Basically, mRNAs are made to be disposable. They're made to have a half-life that's, from, hours to days at the most. And this is important for a few reasons. The first is it allows the cell to respond to environmental stimuli. It also prevents viruses from gaining a toehold in the translational machinery, right? So if an mRNA is super stable and a virus comes in and delivers its nucleic acid, all of a sudden, there's no way for the cell to actually clear that viral infection. But because mRNA is turned over so rapidly, you can actually protect against these viral infections, as well as respond to, say, it gets really hot outside or it gets really cold outside. You're going to want different proteins to be produced based on those environmental conditions. And so one way that this happens is by decapping. And so what you can see here is a co-crystal structure of the decapping complex. The decapping complex actually cuts the inverted 7-methyl-G cap right at this position between the alpha and beta phosphate. You can see here the inverted 7-methyl-G is bound in the pocket, and basically you've got a cut or a cis phosphodiester bond here between 1 and 2, or alpha and beta, in the RNA strand that's bound. And so 5' monophosphates are actually the substrate for an enzyme called XRN1. And this enzyme is always on the prowl in the cell, trying to destroy and degrade all the RNAs it can find. When you have that inverted 7-methyl-G cap, XRN1 can't bind and it can't clean out, right? So here's a co-crystal structure of XRN1 bound to an RNA that has the 5' monophosphate. And you can see that this is perfectly bound by the enzyme. And once you have that 5' monophosphate, XRN1 is processive and just races down in a 5' to 3' fashion, chopping up every nucleotide as it goes along. And this is part of kind of the whole recycling mechanism of the cell to take those nucleotides and then make new RNAs out of them. And it keeps the RNAs kind of in a state of equilibrium. So how do you actually install this cap? Well, in the cells, naturally, this is installed post-transcriptionally. I'm going to talk about some interesting innovations recently about doing it co-transcriptionally, but in a cell, you've got your standard RNA production and then another set of enzymes that come and add the cap on. Well, in the early days of mRNA development as a pharmaceutical, typically you would use the vaccinia capping enzyme to install this cap post-transcriptionally. So here's a quick video showing how it works. You've got your 5' triphosphate from your transcription. The first step of the enzyme cleaves off the gamma phosphate. In the enzyme, you have an active lysine, which then interacts with GTP to form a guanylylated lysine, which then gets used to kind of push together with the 5' cap, the 5' end of the mRNA. And then you've got a methyl donor from S-adenosylmethionine to finish the reaction off. What's not shown here is the next step for cap1, where you would use an S-adenosylmethionine to also add a methyl group to the N+1 2' hydroxy position to get a cap1, which is very important. Obviously, we talked about before for good amounts of protein translation without a bunch of immune reaction. Okay, so what has happened recently is some innovations in the space for capping. So if you're going to make a pharmaceutical out of mRNA, you really don't want a bunch of unit operations. And so as you're making your mRNA and you realize you have to put a cap on, you're going to have to do your transcription, you're going to have to clean up the reaction, then you're going to have to do another enzymatic treatment, and then you're going to have to clean up that reaction. And all of these unit operations results in a loss of purity, a loss of yield, and increased costs and time. Well, back in the early 2000s, there was an invention called ARCA, anti-reverse cap analog. And this was an innovation at the time to actually install the cap during transcription. And so the way that it worked is you would add in this dinucleotide into your in vitro transcription effectively in place of some of the GTP in the reaction. So, let's say that you had, 5 millimolar GTP in your reaction, you would say reduce it down to 0.5 millimolar GTP and put 4.5 millimolar ARCA in it. And this would necessitate initiation off of the ARCA dinucleotide by the enzyme to result in 90% of the transcript being made with the cap already installed, right? So you actually would grow the new mRNA off of this 3' position. And so one of the clever tricks that the team that invented ARCA utilized was actually putting a methoxy group at the 3' of the 7-methyl G, or the inverted G portion, because realistically speaking, this is a fairly symmetrical molecule, and T7 would equally initiate off of the N7-methyl or the regular G. And so if you had the N7-methyl at the N+1 position and the regular G at the inverted position, you wouldn't get translation. So 50% of your mRNA would already be dead on arrival. And so putting this 3' methoxy prevents T7 from initiating at this part. That's why it's called anti-reverse cap analog, because they used to make cap analog where you'd have N7-methyl G here and a regular G here, but you'd only get 50% in the correct orientation incorporated. The other issue with ARCA is that you've got this 3' methoxy, which is unnatural. And so that's a little different, right? If you're trying to make an mRNA medicine, you kind of have to figure out if that's safe or not. But the other thing about it is you don't get cap1 during co-transcription. And as we talked about before, the importance of cap1 for preventing innate immune reaction, or recognizing it as a foreign mRNA, is super important for getting a lot of protein made in a very low immunogenic background. And so if you're going to have to go and do an enzymatic step to install that cap1 anyway, this compound didn't really take off in the manufacturing space for mRNA medicines because it didn't really save all that much time. And it also didn't really save all that much money because you still had to buy this dinucleotide. And it was an expensive endeavor to then also buy the enzyme to put the S-adenosylmethionine donated methoxy on this 2' position of the N+1 nucleotide. So a few years ago, an invention actually came out from TriLink, which is really fascinating. And this is their clean cap product. And basically, they leveraged a few tricks that they learned to actually force initiation with a cap1 nucleotide, or in this case, a trinucleotide, during initiation. And so what you'll notice here is you have an A at the first position, whereas in ARCA, you had a G at the first position. And this is really important. T7 does not like to start off of A's. It's just very bad at it. And so you would essentially make a tiny, tiny amount of RNA product if your DNA templated if your DNA template encoded an A as the first transcribed nucleotide. T7 doesn't like it. It much prefers G. Well, the trick here that TriLink discovered was that if you put an A at that first position on the DNA template, and you then give the enzyme this trinucleotide, where these two nucleotides here allow the enzyme to effectively skip that first nucleotide in the DNA template and start at the second templated nucleotide, which is a G in this case, you get fantastic yields, fantastic initiation because it's starting off of a G. But you also can add significantly less of this trinucleotide because the amount of uncapped material is very, very small, because uncapped material would by definition start with an A. So what a really cool invention here. It allowed you to effectively do a Cap1 installation co-transcriptionally. And so this is actually the compound, or very similar to the compound used by BioNTech and Pfizer in their COVID vaccine to do co-transcriptional capping during the manufacturing of their mRNAs. So what about cap-less translation, or not needing a cap to actually initiate translation? So I'm going to talk about that now. It's basically utilizes a concept called irises, or internal ribosomal entry site translation initiation. So viruses have actually evolved a way around cap-dependent translation. And so this has actually become very important in the circular RNA field, because by definition, a circular RNA does not have a cap because it does not have an end on the 5' region of the mRNA. It's a circle. So how do you actually get translation to occur on a circle? Well, you can utilize an iris. And so, there's really cool things that viruses have learned or evolved really to take advantage of how translation works. And so what you can see here in cap-dependent translation, so the normal M7G type translation is 4E binds the inverted 7-methyl G, like we showed the crystal structure earlier. They have all this other complexes that all come together and it recruits everything, and then boom, you start translation and you start making protein. Well, in this case, you can see this structural element in the RNA actually mimics all of the different components that bind the mRNA in the cap, right? So this is really cool because this allows you to make a circular RNA that can translate. And what we have seen now from a few different labs and a few different companies is that these circular mRNAs seem to have longer half-lives, longer productive half-lives than linear mRNAs. And so basically there's 4 classes of irises. Class 1 and 2 mimic cap-dependent initiation, essentially by recruiting all of the different protein complexes required for initiation. It's basically like an aptamer, right? These structural elements in the RNAs are good binding sites for the protein complexes required that would have normally bound the N7-methyl G. The other 2 classes exploit ribosomal dynamics and structure to affect translation initiation, basically short-circuiting the way that the ribosome actually functions and moves when it's actually going to initiate and make protein. So here's a beautiful crystal structure showing an iris that's actually folded into a structure that mimics a tRNA. And that's just so cool, right? Because basically you've got this RNA structure that mimics all of the different components of a fully functional mRNA with a cap being initiated and translated off of. Okay, so let's talk about RNA sequences. Well, the untranslated region, or the UTRs of mRNAs, have turned out to be incredibly important for changing the amount of protein made per mRNA. And so what you can see here is an example of a UTR evolution at Moderna. And here's the reference, the paper reference that basically talks about how this was done at the company. But basically using a deep learning and genetic algorithm, we've improved our mRNA yields just by changing the UTR sequences 11-fold in 4 years. And so a number of companies have actually come out and showed that improved amounts of protein per mRNA can be achieved by UTR optimization. And it makes sense, right? UTR elements are very important for regulation. They're very important for ribosomal scanning and then initiation. And so there's a lot of parts to the puzzle that can be modulated by changing the sequences that you use in your UTRs. And so it's really just, fascinating to see that, over such a short period of time, such an improvement in total amount of protein made can be achieved just by changing nucleotide sequences in non-translating regions. So let's talk about how to actually get to the translation, right? Get to the translating regions. So on the left here, you'll see is a codon wheel. And so one thing that I really like to point out is, there are known, there are 10 to the 78 compare 10 to the 78 to 10 to the 82 atoms in the known universe, right? If you were to say, take a 300 mer protein, right? So 300 amino acids, and the total codons that are available to code for each of those amino acids, all the permutations would be 10 to 100 to 10 to 150, right? There are literally more mRNA sequence options for a 300 mer protein than there are atoms in the known universe. And that's just an incredible amount of space to search for determining which codon set is the optimal. So let's go through and learn how translation occurs, and we can talk about that some more. So here's a small mRNA that has codons laid out that I'm going to translate. And so first thing you do is you find your 5' cap, and you know that translation will initiate. And you should also look for a 3' tail, 3' poly A tail, and make sure you have both, because you need both to make protein. And we look for the first triplet codon. In this case, it's going to be AUG, but we've skipped ahead, right, to make an mRNA that we can talk about that actually makes a protein that we can discuss. So we have a GAG, right? So G, you go to the center of the codon wheel and you find the G. So we've got a G here in blue. And then we've got A. You stay in this quadrant and then you look for the A, and in this case, it's in pink. And so we've got the G and then in the A, and then the next nucleotide is another G, right? So we've got a G, we've got an A, then we look in this area here and we can find another G. And so that means the GAG codon codes for glutamate, or E, right? So we'll put an E there, right? We've got GAC as the second codon, right? So we've got G, we've got A, we've got C, right? So aspartate is the second amino acid. If you go through and do all of these, it turns into the Edward peptide with a stop codon at the end. So here's a second mRNA. And if we look at the sequence, we can see the first codon is different than the one above, right? GAA. The second codon also different. Third codon is the same. Fourth codon is different. Fifth codon is different. Sixth codon is different. And then actually the stop is different also. So you might ask yourself, well, they code for different proteins. Well, no, they don't. If you actually go through and decode this mRNA into protein, you can see it actually makes the same peptide. And so this is a really important point, right? Because the next question typically is, well, which is the right one? How do you know which one to use? Well, we've made a lot of progress in the field for being able to de novo codon optimize a specific protein sequence. But it's really hard to know exactly which one will make the most protein, because sometimes also you don't want to just make the most protein as fast as possible. Sometimes you want to control the kinetics of protein expression, right? Sometimes you want lower total Cmax, right? But longer duration of expression. Well, you can codon optimize your mRNA to get those different profiles of expression. And so this is really important for different uses, right? So in a vaccine product, you may want a different profile than, say, an enzyme replacement therapeutic. So now that we've kind of learned the rules of mRNA biology, how can we bend them, right? Or how can we actually change the chemistry and change the biophysical characteristics of the mRNA to make a better medicine? And so here's a schematic of an mRNA. And what you can see here is that closed loop formation where the poly A tail is being bound by some proteins, in this case, poly A binding protein, or PABP. They bind to the poly A tail and they interact with the EIF4E and G complex, right, as part of translation initiation. And so, as part of a pharmaceutical product, the poly A tail is typically encoded in the DNA template, but in the cells, it can be added. In cells, it's added by an enzyme called PAP, or poly A polymerase, and that just adds a whole bunch of A's all in a row. In a controlled process, you typically don't want that because it's hard to get a defined number of A's. And so putting into the plasmid template is actually much more effective at getting a tighter distribution of poly A's. So how would you actually start thinking about stabilizing an mRNA to make it last longer in a cell, right? To avoid the standard way that all of these mRNAs get degraded by the machinery in the cell. Well, one idea is to slow down deadenylation, right? Because that's one method that RNAs are degraded, by trimming on from the 3' end, or chewing from the 3' end. The other is decapping, right? And so one thing that Moderna and others have done is actually put an inverted deoxythymidine on the 3' end. So kind of taking a page from the siRNA stabilization field, putting this group at the end of the mRNA actually seems to slow down degradation quite a bit. And so you can actually see a few-fold improvements in total protein output from an mRNA with this type of modification on it. The other idea is stabilizing the cap by using, in this case, phosphorothioates, another hack that's known from the siRNA field that prevents a standard degradation processes, specifically hydrolysis or transesterification of the phosphodiester backbone. Phosphorothioates slow that down. So if you stick a phosphorothioate in the middle of the cap, you actually can slow down decapping. This is another interesting area where maybe there's other chemistries available that can change the dynamic process of decapping also the same way beta-thio would. So here's a slide on genetic circuitry and mRNA logic gates. And so this is a really interesting approach that a lot of companies have started using, specifically to control where mRNAs are being expressed. And so through steric inhibition of translation using tight and specific binders, you actually can change how much of this output protein is made. So L7AE is a protein that binds a specific RNA sequence. And if you put that specific RNA sequence into this mRNA that has this output protein encoded in it, you can actually control where this protein is made. So, for example, in hepatocytes, microRNA 122 is a very common microRNA. And what you can see above here is through microRNA binding complementary sites, you can actually control how much of this mRNA is being degraded and how much of this L7AE is being expressed. So in a hepatocyte with microRNA 122, if you have the complementary binding sites here and you delivered these two mRNAs, you would prevent L7AE from being expressed because the mRNA that binds those microRNAs gets degraded, which would mean you wouldn't have this protein made, which means you wouldn't be inhibiting translation of your output protein. So boom, there, you found a way to deliver two mRNAs, and you found a way to actually control where it actually gets expressed. This is really cool. This is a mirror-on idea. The other approach is essentially a standard mirror-off, right? Where you have an mRNA that you don't want to be expressed in a specific place. And so this paper here talks about a standard LNP, which typically goes to the liver, versus lipofectomy, which typically goes to the spleen. And so what you can see here is mirror-off allows for biochemical biodistribution control. And so if you put a microRNA 122 in an mRNA that goes to the liver, you can see without those microRNA binding sites, you get a ton of protein expression. In this case, it's EPO. But with microRNA 122 binding, it actually gets degraded before it can actually translate protein because this LNP is hepatotropic. If you take that same mRNA for encoding EPO, right, and you put with lipofectomy, you formulate it with lipofectamine, without microRNAs, it goes to the spleen and it gets expressed normally. But if you put 142 binding sites or microRNA 142 binding sites in that mRNA, it gets shut down because those microRNAs are specific for the spleen. So it allows you to actually have some really cool control levers to determine when and where an mRNA is expressed. And I can only imagine how interesting the future is going to be with this idea where you can really start doing computational type controls with mRNAs being delivered to different cells. So let's kind of take a step back and let's think about the mRNAs that can be used for different purposes, right? So what I have here on the left is an analogy of the different ways that the impurities of an mRNA impact how much protein is made. So what I'd like to draw your attention to here is the habanero, the jalapeno, and the bell peppers, right? So habanero peppers are typically very spicy, right? And this is basically in an mRNA analogy, I assign this to mean high levels of double-stranded RNA. We're going to talk about that in a second. Jalapenos, medium, bell peppers, low, right? And so when a cell is infected with a virus, you basically trip two nucleic acid alarms, one that detects uridines via endosomal detection, and one that detects double-stranded RNA in the cytosol. And so uridine detection throws the brakes on protein production. And actually, double-stranded RNA does to an extent, but it's more tunable. So full stop. The problem here is you can't reduce uridine to zero. right? You can change the level, like the amount of them, where they are, and how, the structure of the mRNA with regular uridine, but you always are going to have some uridines. Therefore, you're always going to have some TLR or endosomal detection. And so pseudouridine, or rather the modification that is used in the COVID vaccines produced by both Moderna and BioNTech and Pfizer, actually avoids the uridine alarm while still being translated. This is magic. This is an amazing discovery. And really, Carrico and Weissman, their seminal paper was fantastic to describe this. So basically, this molecule, pseudouridine, is somehow not recognized by the endosomal sensors, the TLRs, but is still recognized by the ribosome as U. And so you can essentially make an mRNA that makes a protein the way a uridine molecule would, but using a pseudouridine molecule, so it actually becomes immune silent. So everyone in the field thought this was going to be enough, but it turned out that you actually needed to control the double-stranded RNA impurities as well. But this is where it really turns into a cool idea. So I like to equate the amount of double-stranded RNA to the spice level in a dish, right? When you're cooking, right? If you have too much, sometimes it's not good, right? So like, for example, these habanero peppers, it's too hot for some dishes, right? You added too much spice, it's not good. Let's say you have an mRNA that has too much double-stranded RNA, it's not good. It shuts down protein translation because the cell thinks it's infected with the virus. You don't get a good response to a vaccine antigen. You can't make an enzyme replacement therapy. It's just not a good product. Well, through lots of efforts at many companies, this was tuned and figured out to how to control it through in vitro transcription process engineering, through mRNA purification, through T7 RNA polymerase engineering. All of these can be used to control and tune the amount of double-stranded RNA in your final product, in your final mRNA. The one thing to keep in mind, though, is that when you deliver an mRNA encoding a vaccine, you want a little bit of heat there, right? You want the immune system to come and check out that cell that's expressing that protein, because you want the immune system to start recognizing that antigen as foreign. So typically, vaccines use adjuvants. So this is a similar idea, but it's not an adjuvant. It's part of the product. And the double-stranded RNA impurities that are part of this process or part of the mRNA product allow you to actually tune or control how reactogenic or how immunogenic your mRNA actually turns out being. And that's very important for a vaccine product and a cancer immuno-oncology product. If you say want to make an enzyme replacement therapeutic, you don't want any immune reaction, right? You want this to be a stealth mRNA. You want it to get into the cell, you want it to make its protein, and you don't want the cell or the immune system to be the wiser. So that's one of the reasons I think the field was able to respond so quickly to the COVID pandemic, was because so much effort was put on removing all double-stranded RNA activation impurities, because most of the field was really interested in controlling all of them for enzyme replacement therapeutics. Vaccines turned out to actually be really in that sweet spot between doing an mRNA in an old school fashion, like making it in an old school way, versus in a very highly pure way, right? Dialing that together allowed you to make a product that had some heat to it, which allowed you to get some immune reaction, which made a very effective mRNA-based vaccine. So let's talk about self-amplifying RNAs, because there's a lot of companies that are now trying this as a way of dose sparing for an mRNA-based vaccine. So a typical conventional mRNA has your linear sequence here, right, with your N7-methyl G cap, 5' UTR, your gene of interest, 3' UTR, and a poly A tail. Well, in a self-amplifying mRNA system, you actually have a very long mRNA that has some additional components in it. And so what you can see here is NSP1, which is right here, is actually an mRNA capping enzyme. And so when you actually deliver this linear mRNA, it starts making these other proteins, specifically NSP1, 2, 3, and 4. And they all work together to actually amplify this negative strand RNA to then replicate it into coding mRNA, which can then be used to make the actual gene of interest subgenomic RNA. Now, the problem here is this self-amplifying mRNA, by definition, has to use regular uridine, so you have a TLR risk. You also have a long RNA, so there's going to be some risk to double-stranded RNA impurities that you can't purify out because of how long the mRNA is. But also, in the cytosol, it has to go through literally a double-stranded RNA intermediate. And that elicits a very strong cytosolic immune response. And so what we have seen in the field right now is that a lot of the companies and academic labs that have tried to bring self-amplifying mRNA vaccines to market see a lot of reactogenicity, or a lot of adverse effects of their mRNAs because they're very immunogenic. And so, basically the idea of self-amplifying RNAs is you can do dose sparing, right? Because one mRNA turns into many mRNAs. You also get longer duration of expression. But again, the cons are some of the issues we just discussed. So one answer to this is actually a trans-amplifying mRNA idea, where instead of just one mRNA encoding all of those pieces, you actually deliver two mRNAs. One that can be made using modified nucleotides to avoid the TLR, innate immune activation, which actually contains the replicase that then replicates the transgene containing RNA that you deliver separately. And so this is really cool because then you wind up getting kind of a linear response in terms of amplification, whereas in a self-amplifying system, it's an exponential response because every replicated RNA then can make more replicated RNA. In the trans-amplifying system, every positive sense transgene that gets replicated is only happening from one of those delivered replicase mRNAs. And so you can actually tune all of this to control it better. So kind of wrapping up the whole story, what are some of the critical quality attributes that are important to pay attention to for an mRNA product? Well, here's some draft guidelines, and here's some CQAs that have been discussed, right? And so for identity, right, sequence confirmation that the mRNA you have in the vial is what it is, you can use NGS, you can use Sanger sequencing, you can use reverse transcription and then PCR. For RNA content, you can also use reverse transcription PCR and UV spectroscopy. For integrity, you can use capillary electrophoresis. For 5' cap, you can do reverse phase HPLC to look at just the first portion of it. This would be coupled with a mass spec. For poly-A, you can do the same kind of thing. Typically, these use RNase H methods that cleave the mRNA pieces off in very site-specific ways. For mRNA integrity, you can use gel electrophoresis or HPLC. For impurities, like specifically double-strand RNA, you can use immunoblots. For residual DNA template, qPCR. Endotoxin, bioburden, stability are standard USP methods, same with appearance and pH. And so I'm going to talk a little bit about LNPs, but this is really an mRNA talk. And so, there will be basically this one slide on LNPs, but I'm available to answer questions if you have more on LNPs. But basically, it's these components kind of mixed together, and you wind up getting a encapsulated mRNA, which protects it from degradation in the blood or in the serum. And also, it allows for effective delivery. And so, one of the interesting things about lipid nanoparticles is that they're in the same size range and have similar membrane composition to viruses. And so they actually get taken up through the same pathways. And this is why it's so important to actually modulate the immunogenic impurities, because it goes through the endosome. It goes through the same methods that viruses are taken up in a cell typically. And so all of those sensors kind of need to be thought of and dealt with or avoided as much as possible. And so the ionizable lipid, as part of the LNP, has a positive charge. It binds to the phosphodiester backbone of the mRNA. The phospholipid and sterols form a kind of a bilayer on the outside to give structural rigidity to the LNP. And the PEG lipid is on the surface here in yellow, is very important for vial stability. So when the LNP dispersion is in a vial, you don't want them aggregating, kind of the way that oil in water, right, will form one large glob, right? You want these little blobs to stay separate. And so putting these PEG, decorating the surface with PEG, actually prevents them from bumping into each other. This also allows for them to be taken up by cells in the body that are specific for immune presentation, specifically antigen presenting cells, or APCs. So having PEG on the surface allows them to circulate for a longer time and allow them to get taken up by the cells that need to express antigens, say if you're making a vaccine. And in the final product, you have some cryoprotectant in the form of sugar or sucrose, and then some buffers to just maintain the pH, because those ionizable lipids need to be kept at the right pH so that they stay very stable and are able to actually deliver the product. So that's the end of the talk. Thank you.
Ed, really wonderful presentation. Thanks so much for sharing that with us. There's some great background information in there and some really great insights into the potential future of mRNA therapeutics. Now, I know the audience is riveted by your talk, that I know for sure, but I know that there have questions as well. Some of them have been coming in already. And if you have a question for Ed, don't hesitate. Just type it into the Q&A box, hit submit. We're going to get to the Q&A session in just a moment and try to tackle as many of those questions as we can. All right, like I said, bear with us for just a moment as we make the transition into the Q&A.
All right, everyone, thanks for joining the Q&A session. We have a bunch of questions here for Ed, so let's get through them and try to tackle as many as we can. Ed, the first question we have, an audience member says, is there any risk for not properly refolding mRNA 3D structure during purification? Would that have an impact on translation or drug efficacy?
Yeah, can you hear me okay?
I can, yes.
Great. This is a great question. You know, one thing that we've noticed, and I think the field has noticed, is that there is definitely a structure that happens upon first pass transcription as well as first pass translation. But we have never noticed a big difference, especially with cap-dependent translation. Now, cap-independent translation has more structural elements involved, specifically the iris, and so you do run the risk of not folding that or refolding that correctly if you do kind of melt and anneal. But in a standard linear mRNA with a cap, we have never seen any difference between a first pass transcribed mRNA versus something that has been melted and refolded and reannealed multiple times. One thing to keep in mind, though, is that melting RNA requires higher temperatures, and that sometimes leads to oxidation of the nucleobases, not just fragmentation of the phosphodiester backbone. And so realistically, you may, by, say, capillary electrophoresis, assume that your RNA is still intact after doing a heat-cool process, but you may have actually damaged the bases that wouldn't show up in that type of method. So, you know, basically, you need to make sure that you're not causing kind of chemical degradation upon heating and cooling, but the structure does not seem to make a difference in terms of translation or drug efficacy.
All righty, thank you for that, Ed. Next question we have, an audience member says, can you elaborate more on how double-stranded RNA impurities are formed in the in vitro transcription reaction?
Yeah, so the double-stranded RNA impurities are fascinating. These impurities are in such low amounts that they're basically impossible to detect through normal analytical methods. Sure, you might see some on a tape station or a fragment analyzer or any sort of gel. You might see some impurities, but you really don't ever get deep down to the double-stranded RNA impurities, which are actually in the sub 0.1% weight kind of mass region. And so these types of impurities actually form by the T7 RNA polymerase, typically kind of jumping back onto the RNA it just made and using that as a template. So you basically have RNA templated transcription, which is really interesting that this happens during the in vitro transcription, right? As you start to build up product in your IVT, the T7 RNA polymerase starts kind of gaining affinity, or rather its affinity starts being matched for using the RNA as a template. And so you wind up actually generating these double-stranded RNA impurities as the in vitro transcription goes along. And there's actually been some interesting work out of New England Biolabs, as well as other groups looking at ways of making RNA by in vitro transcription where you don't make these double-stranded RNA impurities. So like, higher temperature IVTs actually make less of this, because if you imagine the affinity between the T7 and the RNA as a template would definitely be connected to the temperature, right? The binding energies available for that recognition, because it's not a normal kind of promoter that T7 would use. And so modulating, how these double-stranded RNAs are formed, or modulating the reaction conditions to kind of change how much are made, has been a really important part of kind of making safer RNA medicines. And so the interesting thing that also has happened is some T7 mutants have kind of popped up on the scene that actually change the energetics of the double-stranded RNA formations to begin with. But one of the kind of most illuminating experiments that I've seen is simply taking T7 RNA polymerase nucleotide triphosphates and mRNA product, so basically no DNA template whatsoever, mixing those together with some buffer and letting that transcription reaction go. You'll actually see a whole bunch of products made where the T7 actually sits down on that mRNA and starts using it as a template, as long as it's at high enough concentration.
All right, thanks for that, Ed. Next question we have, an audience member says, can you discuss why one would prefer reverse phase HPLC over capillary gel electrophoresis?
Yeah, I think one of the kind of advents analytically in the field has been the discovery of lipid adducts with the mRNAs. And this, again, very, very, very low amount of this impurity actually showing up in an mRNA product that are basically invisible by gel electrophoresis, especially, and capillary gel electrophoresis. Basically, those methods don't typically pay attention to the hydrophobicity of the molecule. They typically pay attention to the charge and the size. And so having a small lipid kind of adduct appended to one of the nucleobases on the mRNA is not going to have a dramatic impact on its gel electrophoresis migration rates. But in a reverse phase HPLC system, you actually do see significant retention time shifting by having a single lipid adduct appended to an mRNA. And so, I think, one direction that the field is moving is the use of these more developed analytical methods to really define what a pure product actually looks like. Because I think one thing that happens in these cases is, you'll define purity and you'll monitor it over time, say in a stability study, and the mRNAs by gel look great. They're super stable, but you wind up seeing a reduction in expression over time, or really efficacy, but if we're talking preclinical work, and that actually, I think, is a perfect example of analytical tools not kind of being up to the task of large biopolymer analytics and single nucleotide resolution analytics, where I think getting a reverse phase system kind of there, looking at the product, is much more informative and actually probably a safer bet.
Cool. Great. Thanks, Ed. Next question we have, an audience member says, can you say more about how to manufacture mRNA encapsulated liquid nanoparticles?
Yeah, sure. So the lipid nanoparticles are the way that most mRNA medicines are administered / delivered currently. There have been some approaches where people use naked mRNA. There's been some approaches where people use lipoplexed RNAs. I don't think anyone has really gone down liposomal formulations, right, with the liquid core. But typically the kind of a solid core lipid nanoparticles, maybe not solid, but I'd say, lipid components, not necessarily liquid components. You wind up getting kind of this mRNA complexed with this lipid ball that looks a lot like a low density lipoprotein. You wind up with a lot of cholesterol typically, and you have this kind of ion pairing with your ionizable lipid to the mRNA. And basically you wind up putting the mRNA into a lipid solution that has all of your lipid components. And, as they kind of crash out of the solution, right, because the solubility of the lipid components in the ethanol just isn't that great. When those precipitate, they basically encapsulate the mRNA as they kind of form these small, lipid nanoparticles. And having kind of the PEG lipids around definitely helps stabilize those dispersions at the particle sizes that you're aiming for, right? So the energy of mixing has a very, very big impact on the final particle properties, biophysical properties, specifically particle size and polydispersity. That seems to be the biophysical characteristics that people pay most attention to. It's an easier kind of bulk measurement that you can use. DLS is typically the way to do it. But so, kind of putting all the pieces together, you then want to remove the ethanol from the solution. Then you want to adjust the pH, because typically the manufacturing process uses acidic conditions, because your ionizable lipid has a pKa typically below 6.8-ish. There's been a lot of great work in the SI field showing the sweet spot for endosomal escape for the ionizable lipid pKa to be 6.5 to 6.8-ish. So mRNA LNPs typically follow the same pattern. And so, removing the ethanol, bringing the pH through the neutral pKa as quick as possible, because if you dwell too long in that neutral pKa, you end up getting kind of accretion of the particles and kind of swelling, not directly swelling, but, growth. Oswald ripening has been one hypothesis floated around for that kind of growth process. But then you kind of go through it, get to a more physiological pH, in 7.5 range, and then it's pretty stable there. As I mentioned in the talk, you'll add some cryoprotectants. Typically sucrose is a common one, but many exist. And the idea here is that it lets you do a frozen liquid formulation. People do lyophilization nowadays of the mRNA LNPs. Those typically require some, buffer and excipient development work. But the standard panels that people use for screening methods for other lyo products definitely are pretty applicable to mRNA LNPs. And then kind of that's your final product. And then there's a whole bunch of CQAs kind of that go along with LNPs. But unless someone had more specific questions, I won't go into that too much.
All righty, thank you, Ed. Take a last look here at any of the questions that have come in, but looking like we're almost at time here. So I think with that, we've come to the end of our webinar. So I'd like to remind the audience that the webinar will be archived on the GEN website at genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues, which we always recommend. I'd like to thank Ed again for his very informative presentation, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to TriLink Biotechnologies for sponsoring this webinar. Hopefully we'll see you again at another GEN webinar in the near future. Goodbye for now. Everyone stay safe and healthy.